Okay, a lot of crowd today. First of all, my name is Euibong Kim. I'm really appreciated you participating in my talk here. And today I will discuss about the introduction to the current drug discovery process. Because I've been working for about 20 years in the pharmaceutical company, and especially I work in the discovery area, which is seeking the drug. And here's a very nice cartoon. It's got like haystacks, and he's trying to find something like a needle. It should be needle, right? Yeah, nails or needles. And it is just like the same analogy we have. And among these haystack, he's trying to find the needle. This is what the drug discovery is. And there's a lot of problem in this process, but I will kind of like introducing today is about the drug discovery process itself. Okay, how the pharmaceutical company. Wow, there's not much test. So everybody likes drugs, huh? Interesting. Okay, I went through process itself, what involved in this kind of process. Okay, and also it's really the great people's enemy is FDA for the pharmaceutical company. How they involve in this drug approval process. And then I will kind of assess what the risk involved in this drug discovery and development. And there's some, is there some place the academia can play the role in this process? And those areas will be mentioned today. So actually, this is the semantic diagram of all process in the one page. So we'll go for like discovery, preclinicals, and then the phase 1, phase 2, and phase 3, and then this part right here in the middle, 1, 2, 3, is the FDA regulations. And then after that, the post-marketing testing is another FDA area. Government is taking place in this area here. We'll go over each of them, what the activity will be involved, and how we get around in terms of the drug discovery process. This one is sad news for everybody. I will get back to this in the last about end of slide. First of all, the area of discovery. So this is a fascinating place. Okay, we can find something just like the needle from haystacks. Okay, it starts from biology department. So we have to have a biologist with us. Without them, we not start the game. Also, there's a lot of people involved in this process. So we have to work another people together, make them to be work together rather than against you. So we have to have a good personality to start with. So usually the choosing disease is the medical field or needed requirement from the society, some disease factors. And those kind of things will be the great factor for choosing disease. But normally, because currently, the pharmaceutical company is involved with very difficult decision making, because the cost of the discovery is taking too much. So they have to have some kind of disease is enough to support their research expenses. That's a very kind of like a choosing disease factor is influenced by that. And also the academia can be contributing in this area. What kind of disease in the problem or have a spread too much in a certain area. We can give them kind of idea to a pharmaceutical company to find the disease. once you find the disease, then the biologist usually find the mechanism of disease. How they start, is it the virus or bacteria or genetic problem, genetic modification, receptor problem, what is involved? And so they study mechanism of the disease. From that mechanism of disease, they can find the target. It can be a receptor, can be an enzyme in certain bacteria, a virus, or humans. So this is the totally biologist miracles for us. So you have to be appreciated how this drug target is defined. It's a lot of people involved with the drug targeting. And first you make the target, then what you do is you're making bioassay from that target. It's a very critical moment, okay? If this bioassay is not working well, then total rest of the process just stop. You cannot do anything, right? So bioassay is important, very important in terms of the studying the drug discovery activity. So you have to isolate enzyme, or you have to know how this receptor is give the signal to the outer body to influencing, what kind of influencing they can do. So first thing what you do is make this bioassay in vitro. That means you want to do in the test tube. Okay? So test tube is very important for us because if it is not in the test tube, we cannot do very much. Right? Then the process to find the drug is going to elongate it long time. It's going to take about 20 years. And then no one going to be interested. Important thing you have to know is the patent law give you right to 20-year exclusive right for 20 years. So if after this patent is expired, everybody can make it. So you spend a whole bunch of money to make this drug. After 20 years, someone else can make it. That means your money is in the air. Your money is go away. We call those kind of activities genetic drugs. Right? As soon as everybody is waiting for the patent expire, so they can make that same chemical moiety, so they can sell into the public. So 20 years is critical time. We have to fight against it. So if you don't have a vitro assay, okay, then the research is gonna be very, very long time. Right? Why is it important? This vitro assay directly related to the high throughput screening. High throughput screening is the vitro assay is in the like a little plastic about 100 hole. Okay? And then they take it into the robotic room using the screen synthetic bank. Usually, most of the pharmaceutical company has millions of the chemicals storage in the frozen. So they already make the solution already. So that's kind of like a robotic arm go to there. One shot is about 100 samples. Take it in and put in the plate to see the activity. So this is kind of like initial screening. Normally, you is screening about million compound. Okay, so million data gonna come into you, and about 10 people or 3 people sit down there looking at the structure. The chemist was like looking at the structure. This point, the chemistry is involved. Okay. So vitro assay, then chemistry starts. So this is a very important, critical moment. That's the reason why vitro assay is so important. Your vitro assay have to be accurate and meaningful. Otherwise, whole total process breakdown. So high throughput screen you're taking about a couple months, screen everything there. Then you can, from this, you can find some this rational drug design technologies can become. So high drug screening is usually involved the synthetic banks. That's the reason why the pharmaceutical companies kind of purchase chemical from everywhere. So academia usually making some special compound, then they can sell into the pharmaceutical company. They're willing to buy all the time. And then another thing is this is the combinatorial chemistry, which is the modern chemistry technology. In one shot, we're making about 1,000 compounds in one reaction. Of course, you have to design the reaction accordingly, but you can make 100,000, 200,000 very, very easily. Okay? So we want to expose all the possible combination to this B2 assay. So we're taking combinatorial chemistry to put the chemical into that assay. Okay? And then another one is the fragment-based chemical. Fragment-based means this terminology is evolving recently because of the drug is kind of like a combination of special ring system. So normally fragmentation is talking about heterocycle kind of compound. In the chemical world, because the younger guys here, chemical world, there's like a lot of ring system. In the biology, our body contains a lot of ring kind of system because ring is very rigid. It's not changing that much. And also, it has a lot of, did you ever heard hydrogen bonding, guys? Do you know hydrogen bonding?
Guys, you love.
In the chemistry, did you ever heard hydrogen bonding?
Think about H2O.
Huh? Yes? What is the hydrogen bonding? H2O. Okay, that's good enough. It's bonding between the oxygen and hydrogen. That's hydrogen bonding. Okay? And then bonding between nitrogen with the hydrogen. That's the hydrogen bonding too. Your DNA is a good example of hydrogen bonding. What's DNA? Hey guys, DNA. Huh? Genetic information, right? Chromosome, you heard the chromosome, right? Chromosome? Okay, chromosome is you've got the chromosome from your father and mother, so you're here right now, right? So chromosome is a typical example of hydrogen bonding. So God using hydrogen bonding all the time. So chemists, medicinal chemists use hydrogen bonding. So this is the fragment is kind of like using this hydrogen bonding a lot. Okay, enzyme use